Welcome to another exciting episode of Interviews. Today we're thrilled to have Mrs. Kristine Manibog, an accomplished biophysicist with extensive experience in gene therapy, computational modeling, and drug development. With over a decade of contributions in the biopharma industry, Kristine has played a pivotal role in advancing the role of gene therapy, particularly through her work with the FDA and Pfizer. Her background in bioinformatics, protein chemistry, and next-generation sequencing has made her a leader in innovative research and drug product development. Kristine, before we dive in, would you like to share anything with us today?
Anything about
Anything about you? Any just general thoughts?
Okay, so I am right now a staff scientist, or they call us staff fellow in FDA, and we are a CMC reviewer. And when I say CMC reviewer, that's chemistry, manufacturing, and control reviewers for drug products, in particular gene therapy products. And gene therapy products are biological products, which is different from, say, what you see in the pharmacy, which is tablets. So that's my current job right now. But in the span of my career after my PhD, I've worked at as a postdoc at Georgia Institute of Technology for just a brief time, and then went on to continue my postdoctoral fellow. It's completely different. And I joined Pfizer as a postdoctoral fellow, and then from St. Louis, and then move here to North Carolina, still within Pfizer, as a senior scientist, and then now as a I call it, reviewer, reviewer of gene therapy products. So yeah.
So obviously a very accomplished person with us today, so it's going to be a great episode. So our first question today is going to be, can you walk through with us your early career and how your passion for biophysics led you to work on gene therapy and computational modeling?
Okay. Yeah, that's a very good question. So let me start first by saying that my major and my research when I was doing my PhD shaped my career of like what I am doing right now. So my research in my PhD is single molecule biophysics, and that's looking at the protein-protein interactions at the molecular level. And I use computational modeling, and I use experimental techniques, looking at how proteins interact in the presence of force. And I transferred that what I see from the experiment to computational. Like I look at the molecular structure in when I say in silico, that's in the computer. So I look at I do experiments in silico or in computer. So that's how my career was shaped. And so when I joined first I joined Georgia Tech as a postdoctoral fellow, starting from protein-protein interaction. I now wanted to be in translational science, and that means looking at living cells. It's no longer just proteins. It's now living cells, looking at protein interaction with cells, living cells, interaction with living cells versus living cells. And these cells are immune cells. These immune cells are very adaptive. They adapt to what they see as antigens in the body. And so because of my research in my PhD and my brief stint as a postdoctoral fellow in Georgia Tech, then I used those skills and the knowledge that I have to join Pfizer. So at first, when I was in Pfizer, I was working on, it's just computational modeling. When I was in PhD, it's both 50% computation, I would say 50% computational modeling, 50% doing experiments. When I was in Pfizer, joining as a postdoctoral, I was only doing computational modeling, looking at molecular interactions of proteins, looking at interactions between small structure and then the protein. And so I look at their behavior. And that's when during that time, there was a huge interest in gene therapy. And then we, like my team, my boss and me and another postdoc, we look into the molecular structures or biological properties of gene therapy products. And when I say gene therapy products, I was looking specifically for adeno-associated virus capsids. So these are vectors or carriers of DNA. So DNA is packaged into these capsids, and then they are used or designed or developed as a product that is injected into the body. And then I was looking into these biological structures. And so that's when I moved to here. And there was an opportunity coming here. And then I again used my experience in St. Louis as a postdoctoral fellow. Coming here, I now do experiments 50% and then computational modeling as a senior scientist first and then principal scientist.
Okay.
So.
And then throughout that whole journey, what really made your interest and motivation peak the highest?
Because it's new, it's very novel for the gene therapy, and it was cutting edge because it's looking at this product being catered into rare diseases, and rare diseases is very hard to cure. Yeah. So they're just treating the symptoms and not exactly the disease. So that piqued my interest. So, and to be a contributor of that is very good for me. So yeah.
And moving on, at Pfizer and now at the FDA, you've worked with cutting-edge gene therapy, as you mentioned. What are some of the key advancements you've contributed to in this area?
Okay, so yes, that's a very good question because unlike generic medicines, unlike the capsules or the tablets that you see in the pharmacy, these are mature products, developed products that has been here for a much longer time than gene therapy. And the gene therapy, because it's a very new product, a novel product, and we are still developing standard or platform technologies to manufacture this product, to analyze or characterize these products. And so when I was in Pfizer, my team and we've developed assays to answer the questions of whether the DNA that is being packaged into what I call these AAV capsids or adeno-associated virus capsids is the real DNA sequence. It's full length or what we call intact. It's the one that when it's delivered to the body, it can treat the patient. So we've developed those assays, and we've used technologies as you've mentioned before, next-gen sequencing technology. We've utilized other technologies in order to completely or fully characterize these products. And it has, in my company before in Pfizer, some units or some teams have adapted the assays or methods that we've developed with me and my team. So
that's really great. So gene therapy has gained significant traction in recent years. How do you foresee its impact on the future of medicine, especially in rare diseases?
Right now, gene therapy is mostly being catered to rare diseases, and there are still struggles because we're still in the process of developing it, maturing to make these gene therapy products as mature as, for example, other medicines that are in the market right now. So what we foresee is that these technologies or these medicines can be designed to now treat chronic diseases such as, for example, diabetes, for example, cardiovascular diseases, so among other chronic diseases. So.
Okay. And you've been involved in both bioinformatics and molecular modeling. How do you balance the computational side of your work with hands-on lab research?
So what I see when you say balance is that I don't foresee this as like it's separate. For example, work-life balance, right? There's a separation between work and life, like outside of work. So in my case, I use the computational side of my research to supplement or to support the observations that I see in my experiments. So, for example, So I've developed an assay when I was still in Pfizer to characterize these gene therapy products, to characterize this DNA. And so we've used this. Okay, so we observe, for example, this is what we observe in the experiment. Like, how can we make sure that this is real? Like, how can we even dig deeper? So we use, again, the next-gen sequencing technology where they look like individual sequences, and we use I use my computational background to develop workflows. I work with a team who has more experience and expertise in bioinformatics, for example, to develop programs to analyze those results from next-gen sequencing. So.
Okay. And then with your experience in quality control and assay development, what challenges do you encounter in ensuring the safety and efficacy of gene therapy products?
So the challenges that I see right now, and even my colleagues see right now, is how we manufacture these products, because again, these are biological products, and so it's very finicky in a way that it's easily degraded. So we make sure that it's not easily degraded. We make sure that there are no residual impurities in those products, because you don't want some impurities that is attached to that product. We make sure that it's safe, it's pure. So that's the challenges we're encountering right now, because they don't have this mature manufacturing process to produce these products and to purify these products. The other one is the analytical assays, or the characterization tests that we do to ensure that this product is pure. So we're still in the process of developing what you call like platform technology, developing products that is say that whenever just a tweak, it won't like give you a very just for example, a very wrong reading, to make sure that the results are consistent. So, yeah.
And you've collaborated across multiple disciplines, from bioinformatics to drug development. How important is interdisciplinary collaboration in the field of biotechnology?
Oh, it's very important. So just I would say, even when I was in PhD, you cannot be a subject matter of expert on everything. So even when you say biotechnology, you touch upon physics, you touch upon chemistry, you touch upon biology. So you have to find people who are experts on those, who are what we call key opinion leaders. So say, for example, when I have this project, and although I do computational modeling, I was not exactly the expert of doing programs, doing analysis for a certain quality or marker of the product. So we've collaborated with a professor who has a graduate student who was doing all this. Again, when I said earlier, we were looking at protein-protein interactions. So he was looking at molecular structures of proteins, or the molecules in a protein. So when we collaborated, we traded expertise, knowledges, and then we put up this project. We finished this project. We published this project, and we were able to publish this project in a more impactful journal, and where there are more people who can be more interested with our work. And even in the industry, you cannot be just a sole scientist doing your work. You have to be collaborative. Again, earlier I said I've developed workflows on the computational side of in gene therapy, and within Pfizer, within the organization, there were multiple experts on different fields. So knowing who to contact, who to collaborate with is very important, so that you can be certain that your project will be successful.
So that's something key, from like even how young you are to how old you are.
Yes.
It's just you can't be good at everything. Like the more skilled people you work with, that's the more skill your product is going to be.
You're right.
Going into around the high school age, for students or young professionals aspiring to a career in biotech, what skills and experiences do you believe are essential to succeed?
So When I say skills, skills like hard skills, for example, technical skills, it's very easy to learn. But what I can say is, because in this generation, you have so many resources. You can look into the internet. When I was young, we don't have a lot of resources. So we have to be resourceful. One, we had to be creative and curious. But now, because you have all the resources, I would recommend your age, or even all high school students who are interested in biotechnology, to just be curious, be curious and be creative in the knowledge that you have. And one practical thing is, if you're interested with one thing, reach out to people. Say in the university, there are many professors who have research in different fields of biotechnology, to reach out to them, because scientists are very willing to contribute, especially like young kids, because they will be the next scientists. They will be the next experts in the future. So be resourceful, be curious. That's very important. Be creative and reach out to those who are experts, because that's where you will learn a lot.
Okay, so like, for example, right, there's probably people like this out there, like a freshman, sophomore, junior, you just found your love for biotech. What is the first thing that you recommend they do?
The first thing that I would ask a high school student, like, what exactly, what field in biotechnology are you interested? Are you interested in making medical device? Are you interested in characterizing drug products? Are you interested in drug development? So those are the first questions that I would ask first, because otherwise biotechnology is a very wide field with very like niche area. So make sure that you know what you are interested in, because you don't want to be a jack of all trades, but then you're not a master of anything, right? So that's the first thing that I would ask him. Like, and then after that, then look into like research. Do some research. If you are very keen, then read books and journals that are into that same medical device. And what medical device do you want to? Like, is it a device for diabetes? The device for say like allergies, the epinephrine. So those are the two things that I would say, or questions first, and then focus on one thing, and don't be like, because you will be overwhelmed by so much information if you can't focus on one thing. So
okay. And I know this is a question that many high schoolers have. In biophysics, how big is physics play a role into that field?
Oh, physics, because physics looks at fundamentals. So for example, before doing my biophysics research, I was in semiconductor physics. And in semiconductor, you look at say structures. Like, for example, your batteries in your cell phones. These are semiconductors, and these are very static structures. Look at the structure, molecular structure. In biology, these are dynamic structures. They move around and they don't stay in one place. So physics, you have these fundamental laws, and then you have this say even Newtonian motion, like F force equals mass times acceleration. That is translated into doing like how does biomolecular structures work or behave, and then the laws in physics are being applied into these biological structures. So I would say, I'm not sure, I think it's still 50-50. I mean, if you are more into biology, you can do that, because there's biophysics degree and there's physics with a biophysics research, and that's what I am. So
so basically just research about your niche, because if it's the physics part that's demotivating you, go into a niche that's less physics, and vice versa. Okay. And then, how do you manage a work-life balance in such a demanding field? And what are some hobbies or activities that you do to recharge?
I have a professor who says there's a time for everything. And because when I was at PhD, I was so overwhelmed with research. I'm always in the lab. But then, are you focusing on your research? Because in 1 hour of being focused on your work, you can do a lot of things. So make sure, because you are burnt out when you don't manage your time. Say you have to block your time. Say even just 4 hours. Okay, I do my work. No social media, no internet, no browsing. And then you focus on your work, and after that you take a break, and then you walk outside, you hike, I do hiking. I like hiking, and then sometimes, I go to the gym and I meet with friends, because you have to be with friends, because friends will, either help you or sometimes tell you, oh, that's wrong, So that's how I balance my work. I have to make sure I have to be. I'm not very good at that right now, so but I have to be very good at. But what I can say is, like, when it's work, it's work. When it's play, then it's play. It's life. So yeah.
And then if you were sent back in time, for example, to where you were a high schooler, what would you do differently to get ahead of everyone else?
Oh, what would I do differently? I would say that I would be more assertive. As a high school student, you're shy and everything. And again, when I was young, we don't have a lot of resources. So I would say, again, when I told earlier, reach out to people who are key experts, I would have done that. Like, ask advice from people who has more experience. Especially, say, if I do research, I will go to, like, a person who has a lot of experience in that research, for example. So that I would change to be more curious and assertive.
So another common trend we've seen on my podcast is, do not be scared to ask professionals for advice, because there's many helpful people like Miss Kristine here. So always be out to get information. Well, anyways, thank you so much for this interview. It means a lot. A lot of people are going to benefit from this. And I'm extremely grateful you came. Thank you.
Thank you.